Today I have the honor of introducing my own mentor, Dr. Gary Bassell. He received his PhD in cell biology from the University of Massachusetts Medical School before conducting a postdoc at Harvard Medical School. He then accepted an assistant professorship at Albert Einstein College of Medicine before coming to Emory in 2005, where he is now a professor in the departments of cell biology and neurology. And you know him as the guy who let you in here, since he is the chair of admissions right now. So please join me in welcoming Dr. Gary Bassell.
Well, thank you, Kristen. And we always get worried when a graduate student introduces you, so she really behaved herself. So what I hope to do today is to make the field of mRNA biology seem interesting to you. For many years, we've actually been interested in the mechanism, regulation, and function of mRNA transport and local protein synthesis in neurons, and how that is important for a number of aspects of neuronal function. Our laboratory is kind of divided into three camps. A group of people who are interested in basic science, basic mechanisms involved in dissecting out the molecular machinery involved in mRNA transport, local protein synthesis, and their function in neuronal development and synaptic plasticity. This research guides our interests in neurologic diseases that result from impairments in the machinery that's involved in mRNA regulation in neurons. And more recently, this research on disease models has guided translational research in our laboratory to design therapeutic strategies that might correct for RNA dysregulation in the nervous system, working in human cells and also in animal models. So let me just pose the question here that was posed 25 years ago. Why do cells localize RNA? Actually, it was originally thought that there really was no reason for RNAs to be distributed to peripheral parts of the cells. It was believed to be just diffusion and not really serving any functional role. However, we now know that RNA localization is a highly conserved biological mechanism from yeast to mammals. mRNAs are sorted in cells, where some mRNAs actually hang around outside the nucleus, and others can be trafficked to distal extremities. In this case, in yeast, the translocation of a specific RNA from the mother cell to the daughter cell can influence budding in yeast and mating type switching. RNA localization in oocytes and in early embryos can lead to the establishment of protein gradients that can influence patterning within the embryo. In the case of neurons, specific RNAs can be transported either to the developing axon and the growth cone, or into dendrites and dendritic spines. And so the talk today is going to focus on two stories. One is the RNA localization to the developing axon and the growth cone, and the second is in the dendrite. What are the molecular mechanisms that are involved in these processes, and how might they go awry in different diseases? So the hypotheses that kind of guide our lab and many others are as follows. The localization of specific mRNAs to distinct intracellular compartments provides a mechanism to sort proteins to sites of function and affect cell function. mRNA localization is mediated by molecular interactions between cis-acting elements, transacting factors, and molecular motors. And more recently, we've been interested in the hypothesis that mRNA binding proteins and associated factors act as localized translational switches in response to receptor stimulation, in particular in response to neuronal activity. So now for the first part of the story. Actually, when I was setting up my laboratory at Albert Einstein College of Medicine many years ago, a key question is whether RNAs were sorted. Could you, show that, one mRNA was confined to the soma and another could be targeted to the periphery of the cell? And using double-label fluorescence in situ hybridization in cultured cortical neurons, beta-actin and gamma-actin mRNA can both be detected in the cell body. But if you look at the distal axon. It's only the beta-actin mRNA that extended into the distal axon and accumulated in the growth cone, where the gamma-actin mRNA was confined to the cell body. We observed these puncta, which we refer to as granules, suggestive of packets that can perhaps be transported down the axon and the growth cone. Over a number of years, in collaboration with Rob Singer's lab at Albert Einstein College, in collaboration with James Zheng's lab here at Emory, we've really dissected out the molecular mechanism of beta-actin mRNA localization to developing axons and growth cones, mechanisms involved in local protein synthesis of beta-actin, and demonstration that it plays an important role in axon guidance. So to summarize some of these studies right here, beta-actin mRNA localization is actually mediated by a zip code sequence that's in the 3' untranslated region of the mRNA. So this is the coding region that encodes for beta-actin protein. But in the 3' untranslated region, this is rich in regulatory information, sequences that can influence where an RNA is transported and whether it's expressed, translated, in response to certain receptor stimulation pathways. This 54 nucleotide zip code sequence is actually not found in gamma-actin mRNA. But if you were to take that sequence and copy it into the gamma-actin mRNA, you would direct that RNA out into the axon and to the growth cone. The RNA binding protein that binds to the zip code we refer to as zip code binding protein. It's an RNA binding protein that has multiple RNA binding domains. But these domains down here at the carboxy terminus, KH3 and KH4, bind very tightly to the zip code sequence. And that binding actually represses the beta-actin mRNA, so it can't be translated. And this is a key feature of transported mRNAs. They are actually translationally repressed en route, because it would defeat the purpose of RNA localization if you were making proteins along the route of RNA transport. The goal is to enrich proteins directly at their functional sites. So ZBP is both a transporter and a translational repressor. The other part of the protein, the amino terminus, actually binds to kinesin. So ZBP is a bifunctional protein, serving as a repressor and serving as a transporter to drive the beta-actin mRNA down to the growth cone. And then, in response to receptor stimulation and activation of Sark family kinase, which is activated in response to neurotrophin receptor stimulation, Sark family kinase will phosphorylate this highly conserved tyrosine that's just upstream of the RNA binding domain. And when that's phosphorylated, that will cause the repressor to fall off, released from the RNA, allowing the RNA to be translated by ribosomes at the 5' end. That precise molecular mechanism is not really well understood, but still this work was really important because it put forth the concept of these translational switches that allow spatio-temporal control of RNA localization. We can actually see that zip code binding protein traffics down the axon to the growth cone and also traffics back to the cell body. When it's trafficking down, it's bringing the beta-actin mRNA down there, and it actually can then traffic back to the nucleus to get more. That's a model that we have that we're currently working on. These particles move very fast. They move at over a micron per second, and they're propelled by kinesin along microtubules. What happens when the RNA gets to the growth cone, though? How can we actually image the process of local translation? And so one way that we can do that is actually to engineer these fluorescence reporters that provide readouts of local protein synthesis. So here we're not looking at the endogenous mRNA, rather we're looking at an RNA that encodes a fluorescent protein. So here the beta-actin 3' untranslated region, which contains the zip code and can target a reporter RNA. The reporter encodes dendra, which is a photoconvertible protein that is fluorescent, green fluorescence that can be irreversibly photoconverted to red fluorescence with UV or laser. And then you can look for the appearance of new green fluorescence, which would be indicative of new protein synthesis. And we can prove that it's new protein synthesis because we can add protein synthesis inhibitors to the environmental chamber and abolish the appearance of stimulus-induced translation. We've also engineered a membrane tagging sequence. So following the synthesis of dendrite, it's going to be anchored to membranes, and it'll be diffusion restricted. So with this reporter, a postdoc in the lab, Christy Welshans, transfected this into cultured cortical neurons, both wild-type neurons and neurons that were cultured from zip code binding protein knockout mice. These ZBP1 knockout mice have a high perinatal lethality due to the loss of ZBP, which is an important protein for development. But we can culture neurons at embryonic stages and actually look at their growth cones and see if they have impaired a local translation using this photoconvertible fluorescence reporter. So what you're looking at here is a growth cone from a wild-type mouse. And here is pre-conversion, here is post-conversion, green converted to red, and what you're seeing here are hot spots of local translation. At 30 minutes after stimulation with netrin, which is an axon guidance factor, an attractive cue that actually stimulates growth cone steering, and we believe that growth cone steering is dependent upon local translation of beta-actin. This is the type of fluorescence recovery that you get when you do this photo conversion and look for the appearance of new green fluorescence in response to netrin. And you can see that wild-type neurons show robust expression, robust local synthesis, but that is abolished in neurons from the ZBP1 knockout mice. They have impaired localization of the RNA, impaired ability to synthesize beta-actin in response to receptor stimulation, which presumably is dependent upon SARC activation and phosphorylation of that tyrosine that I showed you. So these are experiments that Christy did showing that a wild-type growth cone will steer toward a pipette, releasing netrin, whereas a growth cone from ZBP1 knockout mice, the axons actually extend normally, but they don't steer toward the gradient because they can't synthesize beta-actin as well as other proteins that are ZBP1 targets that we're currently studying. This is the quantification of that data right here, looking at steering responses. I should point out that a lot of this work, the earlier studies were done in collaboration with James Jang's lab, where we actually first observed this on Xenopus neurons, and then his expertise was very important in moving this to the mouse neuron system, which allowed us to look at a genetic null of ZBP1 and show that there's impaired steering responses to netrin when the growth cones lack ZBP1. Yet the axons can grow. So in the absence of ZBP, the axons actually grow, but they can't steer. Remember that I told you that we were interested in the role of these phosphorylation switches. So here's the switch that I talked about. ZBP transports the beta-actin mRNA in a translationally repressed complex. It gets to the growth cone, and then in response to receptor signaling, ZBP is phosphorylated by SARC, and then it will fall off the RNA, removing translational inhibition and allowing for local protein synthesis of beta-actin to affect actin dynamics on the near side of the growth cone, allowing for steering. I showed you that neurons cultured from ZBP1 knockout mice have impaired steering. Similarly, if you overexpress a dominant negative, a non-phosphorylatable ZBP1, where a tyrosine is converted to a phenylalanine, this locks the ZBP on the mRNA, rock solid, so it can't respond to the cues. Beta-actin mRNA is translationally repressed. In this phosphomutant, the ZBP can actually traffic to the growth cone. This is a very useful reagent because it allowed the dominant negative allowed us to actually uncouple the role of ZBP in transport from its role in translation. And so in this case, where phosphorylation was prevented, you had impaired turning responses to netrin, similar to the case of a ZBP1 null. Yet the axons could grow normally. So outgrowth was not impaired with the dominant negative, but steering was impaired toward netrin. So it mimicked the effect that we see in ZBP1 knockout. So over the years, we really have a kind of, in collaboration with several labs, really refined our understanding that RNAs can be sorted. Some mRNAs stay in the cell body, others move out in these particles down microtubules propelled by kinesin. They're translationally repressed, and then in response to axon guidance factors, you can have activation of kinases that can remove translational inhibition and allow proteins to be made on site, on demand, in a spatio-temporally controlled manner. But there's really much research to do in the coming years. We want to know if this mechanism functions in vivo. Is it required for axon guidance in the developing nervous system in vivo? And we have collaborations on that project going on. We want to know if ZBP does the job alone. It's kind of unlikely that you have one RNA binding protein that was solely responsible for transport and translational regulation. And we're identifying other RNA binding proteins that are in these particles that we refer to as granules. Interestingly, one of the proteins that we were studying early on is called zip code binding protein 2, which also plays an important role in this process. I'm actually not going to talk about that, but what I will tell you is, sometimes you can actually be studying one protein and its partner turns out to be more interesting. And we were really kind of seeking an example, a disease model. We were seeking an example. Our prediction was that if these mechanisms really are important for neuronal development, that there would be inherited neurological diseases that result from the failure to express proteins that are important in either the transport or the translational regulation. And so it turned out that one of the proteins that we got really interested in was the survival of motor neuron protein, because it was shown in a yeast-2 hybrid assay to actually interact with this CBP2 that I didn't really talk about. But we got really interested in survival of motor neuron protein because it is deficient. Its deficiency, its inherited deficiency, leads to this autosomal recessive motor neuron disease called spinal muscular atrophy. This is a neurodegenerative disease that affects primarily motor neurons, and it's caused by mutations in the survival of motor neuron gene, encoding this ubiquitously expressed survival of motor neuron protein, which has a well-described function in SNRP assembly. SNRPs form the spliceosome, which is involved in splicing. But it was really unclear as to how loss of such a housekeeping function that was required in all cells in the body would lead to a neurodegenerative disease affecting primarily motor neurons. Might SMN fill some non-canonical role in motor neurons, perhaps related to RNA localization or local protein synthesis? So we got very interested in this. It's a very clinically relevant problem because SMA is the most common inherited neurologic cause of infant death. So interestingly, SMN is transported, like ZBP, in axons and motor neurons. All of the work that I'm going to talk about on our SMN and SMA story is done in collaboration with my colleague Wilfred Rossell, who's an assistant professor here at Emory, and he actually brought the ability to work on motor neurons here to Emory. And what you want to do is you want to study the type of cell that is affected in the disease. And so we've been able to actually look at motor neurons, and in particular motor neurons cultured from transgenic mouse model of spinal muscular atrophy. The survival of motor neuron protein, like zip code binding protein, forms these particles in axons. They're actively transported in axons, and biochemically they associate with the RNA binding proteins that we have been studying. For example, zip code binding protein actually has a strong co-immunoprecipitation interaction with the survival of motor neuron protein. Yet when you delete the tutor domain, which is a hotspot for missense mutations that cause SMA, spinal muscular atrophy, you have a significantly impaired interaction with zip code binding protein. So we imagine that SMN, in addition to its role in SNRP assembly, actually plays a role in interacting with these RNA binding proteins that are important for mRNA localization and local protein synthesis. And Claudia Fellini, a postdoc working jointly with Wilfred and me, had some very striking observations that if you culture neurons from spinal muscular atrophy model mice, or if you use short hairpin RNAs, this is the data that's actually shown here. If you use short hairpin RNAs to silence SMN, there's impaired RNA localization in the axons of motor neurons. And we actually think that that may play an important role in generating the axonal dysfunction in SMA. Actually, in SMA, you have a number of examples of axonal defects, including this dying back axonopathy that occurs both in the mouse model and in human patients. So this is the poly-A signal in a wild-type neuron, and this is the poly-A signal in SMN-depleted neurons, and you can quantify that by quantitative digital imaging analysis. More recently, Claudia Fellini and Paul Donlin, a new graduate student in our lab working jointly with Wilfred, is working to identify what are those RNAs that are localized. Wilfred early on showed that beta-actin mRNA localization is dramatically impaired in growth cones and axons from SMA model mice or in SMN-depleted neurons by short hairpin RNAs. Claudia and Paul are now very keen on investigating impairments in GAP43 mRNA localization. So GAP43, what is GAP43? It's growth-associated phosphoprotein that plays an important role in the actin cytoskeleton, important role in axon growth. It's a neuron-specific protein, so it's a really attractive candidate for a mislocalized RNA playing a role in the axonal pathology in SMA, and that's something that we're actively studying. Actually, a prediction is that if RNA localization is impaired, that would lead to a depletion of the encoded protein. So it's insufficient really to just show that the RNA localization is impaired. You have to really do immunofluorescence and other types of methods to show that the protein localization is impaired. So here, GAP43 immunofluorescence shows a reduction in motor neurons. These are actually motor neurons cultured from spinal muscular atrophy model mice that also have a high lethality postnatally and die shortly after birth. So in terms of this story right here on SMA, we really think that the survival of motor neuron protein may serve this non-canonical function in RNA localization, where it doesn't bind RNA directly. Actually, SMN by itself has very weak RNA binding activity directly, but it does bind strongly to RNA binding proteins. So our model is that SMN helps RNA binding proteins do their job. It plays a role in perhaps the assembly of a complex that is competent to be transported down the axon by active transport. That's a model that we're currently, exploring. I should emphasize that we really don't have evidence for reductions in the steady state levels of RNAs in cells upon SMN depletion. So if you look at beta-actin and GAP43 mRNA, at least in experiments where you're silencing SMN by short hairpin RNA, their total expression of the RNA is not affected. It's their localization into the axon. We have some, we're still sorting this out, but it really seems that SMN plays a role in RNA localization. So that's going to complete the story about the axon. For those neuroscientists that are really interested. So in our lab we have two camps. People that are more interested in the axon, and then another group of students and postdocs that are more interested in the dendrite. A disease that we think results from impairments in dendritic protein synthesis is Fragile X syndrome. And I'm going to talk a little bit about research that we've done over the years on trying to study how this protein that's deficient in Fragile X syndrome, FMRP, plays an important role in RNA localization and local protein synthesis. So Fragile X syndrome is the most common inherited form of intellectual disability. It's also the most common monogenic cause of autism. Thus, research on Fragile X syndrome has really important implications for understanding autism. And I'll talk more about the shared neurobiology between Fragile X and a number of forms of autism spectrum disorders. This is an X-linked disease, which means that it's going to affect males more commonly than females. This young man has intellectual disability, and he has also a seizure disorder. This young lady here does not have intellectual disability, but she does have some features of autism. She actually does. This particular young lady does not have meet the diagnostic criteria of autism, but she has mood disorders, she has anxiety, and she has some seizures. Actually, so intellectual disability, of course, is the most common phenotype symptom. Developmental delays, learning disabilities, hyperactivity, attention deficit disorder, anxiety and mood disorders, and seizures. We are very much interested in how the loss of FMRP leads to these impairments, presumably in neuronal function, synaptic plasticity. Fragile X is caused by this trinucleotide repeat expansion in the FMR1 gene that leads to gene silencing. So in this disease, it's a loss of function disease. It's the absence of the encoded RNA binding protein that leads to these signs and symptoms. So what is FMRP? Well, like ZBP, the protein I talked about earlier, it has these KH domains, which stands for K homology, homology to the protein HNRNPK, which is the parent member of this family. It also has an RGG box, an arginine-rich sequence. Actually, the RGG box binds to many RNAs that have this G quadruplex structure, a G quartet structure. FMRP binds to about 3 to 4% of RNAs in brains, but this is a biologically coherent data set. Okay, so these RNAs that are recognized by FMRP encode proteins that are really important for neuronal development and synaptic function. And many of these RNAs have this molecular structural motif, this G quartet. The RNA binding protein FMRP shuttles in the nucleus where it forms a complex with the RNA and transports it out into the dendrites and localizes to dendritic spines, where it associates with polyribosomes. We and others are interested in how FMRP regulates translation of target mRNAs in synapses, in particular at spine synapses of glutamatergic neurons. Why are we interested in dendritic spines? Well, it's known that in dendritic spines you actually have polyribosomes. And it's known that specific mRNAs are transported into dendrites. And polyribosomes within spines are believed to play an important role in activity-dependent refinement of synaptic connections and the maturation of dendritic spines. In the neuroscience courses here, you actually learn a lot about how spines, which are the sites of excitatory synaptic innervation in the brain, these are predominantly glutamatergic excitatory synapses. You've learned that experience-dependent changes in spine morphology underlie long-term synaptic plasticity, important for learning and memory. Sculpting, pruning of spine synapses is important for learning and memory. What I want to add to this is that gene expression is critical for this process. Many of you are familiar with transcriptional regulation in the nucleus, but you also have gene expression at the synapse. If polyribosomes are out there, that allows for on-site, on-demand local protein synthesis to influence spine morphology, which will in turn influence synaptic strength. Many of you know from the courses that we've taught in neuroscience that plasticity is input specific. How do you achieve that input specificity? How does one synapse, how is it strengthened independent of its neighbors? Protein synthesis is directly required for this process, local protein synthesis. Actually, if you prevent local protein synthesis, you affect the ability of neuronal activity to sculpt and shape spines. And the absence of protein synthesis can lead to impaired learning and memory. So is Fragile X syndrome due to the loss of FMRP-mediated regulation of local protein synthesis? That's what we really want to ask. That's the hypothesis, that FMRP mediates the regulation of mRNA transport and local protein synthesis, important for synaptic protein localization and signaling. And in the absence of this, you have a dysregulated neuron that can't control where RNAs go and how their translation is regulated in response to activity. That leads to the impaired plasticity, the impaired spine morphology, the cognitive dysfunction in Fragile X syndrome. That's the hypothesis that we in so many labs are looking at. So our laboratory, in collaboration with Steve Warren and others, have really helped to tease out a number of functions for FMRP. Transport function, translation function, signaling function. I'm going to really focus right now on the translational switch. Remember, I talked at the very beginning that a hypothesis is that RNA binding proteins like CBP are parts of a multimeric complex that provide a translational switch, allow proteins to be synthesized on demand in response to cues from receptor stimulation. So in order for a candidate RNA binding protein to play a role in learning and memory, it must be able to turn on and off translation in a highly tuned, spatio-temporally defined manner. And we believe that FMRP is one of those such molecules. So in collaboration with Steve Warren's lab, we know that when FMRP is phosphorylated, translation is shut off. That's the opposite of ZBP. So here we have a dephosphorylation-induced switch. So in response to activation of glutamate receptors, in particular metabotropic glutamate receptors, you activate this phosphatase very strongly, PP2A, dephosphorylates FMRP, and allows for synthesis of proteins that affect synaptic plasticity. And one of those proteins is PSD95 that I'm going to be talking about. And I wanted to tell you about how we think this mechanism is actually reversible and how it is regulated by receptor signaling. And then I want to talk about what happens when you don't have FMRP. And the neuron really can't make proteins on site, on demand. How does that really affect the ability of a neuron to respond to receptor stimulation, and what are those broader consequences of FMRP deficiency? I'm talking here to graduate students, so I thought it would be really quite appropriate to actually tell you that the research in our laboratory began by a graduate student, Laura Antar, who's now an assistant professor in psychiatry at Albert Einstein College. But she actually started up the FMRP project in my lab. We weren't working on FMRP before, and she discovered that FMRP was actually localized to dendritic spines. It was known that FMRP could be in spines by EM, but the prevalence was not really appreciated, and she did a lot of quantitative imaging where she showed that FMRP granules shown in red. This is a 3D reconstruction of a dendritic spine. In green, you're looking at F-actin, filamentous actin. Spines are actin-rich. The red is the FMRP particles. The blue is the presynaptic glucan. And you can see the FMRP going out into the spine head. Importantly, FMRP associates with RNAs other than beta-actin. So these are distinct from zip code binding protein granules. These granules contain RNAs that encode important components of the postsynaptic scaffold and spine, like CAM kinase and PSD95 that I'll talk about. So what is PSD95 and how does FMRP regulate PSD95? So PSD95 is the most abundant component of the postsynaptic density. And you know from your coursework that spines have very complex molecular architectures. They have these ensembles of proteins, of scaffolding proteins and transducers that transduce information from receptor signaling to downstream cellular kinases and also affect the cytoskeleton. This is really very confusing, but you see here that PSD95 has these modular PDZ domains that many proteins, scaffolding proteins have, but they allow you to actually link molecules together in these ensembles. So where you have the yellow circle here, those are examples of PSD95. So it's a real critical molecule at the synapse. It's believed to play an important role in synaptic strength. It also plays an important role in synapse development, spine morphology. It interacts with so many interesting molecules that we were really actually excited to find out that it's localized to the dendrite and that FMRP associates with PSD95 mRNA. That was work from Ravi Mataschetti, a postdoc in our lab, and also discovered by an Italian group, Claudia Abani, the same year. What Ravi Mataschetti has done in our lab is to reveal a mechanism for this translational switch. So FMRP actually partners with microRNAs to reversibly regulate mRNA translation in a phosphorylation-dependent manner. What are microRNAs? Well, microRNAs, like FMRP, are regulated breaks of translation. Many mRNAs have regulated breaks that act on them, either RNA binding proteins or microRNAs, which are small non-coding RNAs that act as breaks. What we showed is they partner together. They actually don't act in parallel, at least in the case of regulating PSD95, but they do so in a concerted complex. MicroRNAs are known to be localized to dendrites and axons. They are impaired in a number of neuropsychiatric diseases, and so we postulate that Fragile X syndrome may actually be a disease of a dysregulated microRNA system. We know from work that Christina Gross did in the lab that microRNAs are localized to dendrites. Several labs have shown this. Nina was the first to show this in vivo. Can also show this in cultured neurons by fluorescence in situ hybridization. We're focusing our attention here on miR-125A, which extends beyond the cell body layer and into the dendritic layer. miR-125A, we've shown, targets PSD95 mRNA and acts as a brake on translation through a partnership with FMRP. So I'm going to show you some data that's published from this paper and about how we think this really provides evidence of a reversible model for the ability to make proteins on-site, on demand, in response to receptor stimulation. So remember I told you that FMRP, when it's phosphorylated, shuts off translation. Well, how does it do that? It does that by partnering with RISC, which is the RNA-induced silencing complex comprised of argonate proteins. Each argonate protein is loaded with a single microRNA, and that microRNA will base pair to the mRNA. And RISC represses translation, so it's a strong repressor of translation. FMRP, when it's phosphorylated, recruits RISC. And so FMRP, we think FMRP is co-opting RISC to silence its transcripts, that it's hijacking RISC, that on its own, it actually, we wonder, on its own it might not be able to repress translation, but when it's phosphorylated, it can silence translation using RISC. And when it's dephosphorylated in response to PB2 activation, that phosphatase, FMRP ejects RISC from the translational inhibitory complex. Remember, in the case of ZBP, it was a phosphorylation-induced switch, where ZBP falled off the RNA. Here, the RNA binding protein doesn't fall off. It stays there, but it's a dephosphorylation-induced release of a binding partner. So it's a different type of a switch, but the same concept, allowing you to make protein on-site, on demand, in response to glutamate receptor activation. And we think this has an important effect on synaptic morphology and architecture. This work was a collaboration with Steve Warren's lab over a number of years here at Emory, and I'll show you some data that support this this model. The first thing that we wanted to do is how do you show that a microRNA represses translation? Well, one way is to make a reporter. And in this case, it wasn't a fluorescent reporter, it was a luciferase reporter that we're transfecting into cultured cells, in this case a neuroblastoma cell line. So we have two types of reporters, a wild-type reporter and a mutant reporter. The mutant reporter has a disabled miR-125A binding site, so it's insensitive to miR-125A inhibition. And we compare luciferase activity, expression, wild-type and mutant reporters. And we actually look at the effects of overexpression of miR-125A. So our model is that miR-125A is a brake on translation, it's a repressor on translation. And sure enough, it can attenuate expression of this reporter, of the wild-type reporter, but not the mutant reporter that has a disabled binding site. Conversely, if you sequester the endogenous microRNA with anti-miRs, you have the opposite result, where sequestering endogenous miR125A. You remove the break on translation, you have elevated translation. So here you have an attenuation, here you have an elevation of translation of the wild type reporter, but not the mutant reporter. But that doesn't tell you anything about FMRP. In FMRP knockout neurons, we actually observed a lot of PSD95. The neurons are making too much PSD95, too much translation, because that's due to the loss of FMRP. The loss of FMRP, which is a brake on translation, which is a negative regulator, it's a repressor of translation. So here, in the control neurons, what you're looking at here is a Western blot for PSD95 in FMRP knockout neurons. So these are cortical neurons cultured from FMRP knockout mice. A lot of PSD95 protein in the control. If you transfect in FMRP into these knockout neurons, putting FMRP back in there to restore repression, you drive down PSD95 protein levels. You can drive it down real strongly when you put in a phosphomimic complex that has very tight binding for RISC. So in this assay here, we're actually looking at the ability of this phosphomimic complex, where the serine is converted to an aspartate. That has, this binding assay shows that you have actually very strong interactions between RISC, the microRNA 125, which is the brake, and the FMRP. And this drives down translation very strongly, and you have very little PSD95, whereas the serine to alanine mutant, which is not phosphorylatable, can't inhibit a translation and actually behaves like an FMRP a knockout. Importantly, the micro is required for the ability of phosphorylated FMRP to inhibit translation. And so here you actually have phosphomimic FMRP driving down the translation, but if you sequester the endogenous mir, you can't attenuate translation. So the ability of phosphorylated FMRP to inhibit translation depends upon the microRNA in the complex. I'll show you one last piece of data that really speaks to this model. A prediction is that if you were to take the Fragile X syndrome model mice, FMRP knockout mice, you'd expect that there would be reduced interaction between RISC and the PSD95 target mRNA. FMRP acts as a bridge to recruit RISC onto the target mRNA, and that's indeed what we observed. So in synaptic neurosomes from FMRP knockout mice, there was reduced interaction between AGO, the RISC component, and the PSD95 mRNA in synaptic neurosomes, as opposed to total lysate. So we think that phospho-FMRP is actually recruiting or stabilizing this complex, this RISC complex at the synapse. And in the absence of FMRP, the prediction is that you have too much protein synthesis in the dendron. But how can we actually observe that in live cells? A lot of what I've told you about that paper that we've studied on how FMRP regulates translation in a phosphorylation-dependent manner, a lot of that is deduced from biochemical means. So Marius Ifram, a real talented postdoc in the lab, who is interested in single molecule imaging, he has co-developed this method when he was a graduate student at UConn with John Carson. And unlike, instead of using a dendro reporter, here he's actually using a venous reporter. So venous actually acquires fluorescence much more rapidly on its own, but it also bleaches very rapidly on its own. So it allows you, with high-speed imaging, using laser-based excitation. Marius is doing this work in collaboration with Ivan Resnick in the physics department. That here you have the PSD95 open reading frame fused in frame to Venus. And we also have a membrane tagging sequence. But Venus is at the amino terminus, so it's going to fluoresce on its own very rapidly, and it will bleach on its own with appearance and disappearance of fluorescence. So within just a couple of minutes, you can actually have fluorescence, and then within a second, that fluorescence will bleach with single-step kinetics. So this really provides for high spatial resolution and high temporal resolution, and Marius has been able to image transfected cells and to look at different areas. So here he's interested in this region of this secondary dendrite. You see a number of dendritic spines, and what you're doing is we're building an event map. So we're looking, for example, this is a very small region, 1 micron by 1 micron, and we're looking at the appearance of Venus, and then the disappearance of Venus, consistent with the synthesis and bleaching of a PSD95 molecule. And we can ask how this occurs in response to receptor stimulation, and build an event map, and then build an event schedule, which is an event map over time, and look at these translational events over time. And here you have the raw data, and here, if you watch, you'll see this is after using image analysis software to fit a Gaussian fit to look at these single molecules. You can see the appearance of PSD95 being synthesized in these dendrites and in these dendritic spines. We know that this is protein synthesis dependent, and we can now look at a Fragile X dendrite and ask what's wrong. Our prediction from the biochemistry experiment is that there's too much protein synthesis in the absence of FMRP, because in the absence of FMRP, you can't recruit risk to the PSD95 mRNA, and then you have inappropriate, excess, promiscuous translation. That's deduced from the biochemistry and from the molecular biology analysis that we've done. And so it's very exciting to actually be able to see this in a dendrite and in spines, that neurons from FMRP knockout mice are indeed making too much protein. But I don't think that, Fragile X is just a disease of too much protein synthesis. You hear a lot about that in the literature, that Fragile X syndrome is a disease of too much protein synthesis, runaway protein synthesis. I think that, importantly, it's also a disease of dysregulated translation, because in these knockout neurons that are missing FMRP, not only do you have too much protein synthesis, the synapse can't make proteins on-site, on demand. So there's a dysregulation, there's an occlusion of stimulus-induced translation. So a wild-type neuron, you can see in response to DHPG, which is an agonist of group 1 metabotropic glutamate receptors, it will stimulate translation of PSD95. However, in the knockout neurons, you have a flat baseline. This is normalized. So actually, what's not shown here is the translation is actually in excess. But what you can see here after this normalization is that there is an occlusion to the agonist stimulation. So if you compare the two data sets, excess protein synthesis at basal states and occlusion of agonist-induced protein synthesis, really suggesting that translation is indeed dysregulated. We're actually really excited to have that data from Marius, because it really starts to put together these pieces of the story. Mark Baer at MIT and Kim Huber, when she was a postdoc in his lab, they took brain slices from FMRP knockout mice and just put S35 methionine on it, okay, methionine that's radio-labeled, and look at rates of protein synthesis. And what they observed is that a Fragile X brain slice made too much protein synthesis. So that really kind of fits with the data that we have so far. Interestingly, if you antagonize the group 1 metabotropic glutamate receptor with an antagonist, you can quiet down that excess protein synthesis. By quieting down the excess protein synthesis with mGluRs, you can actually correct Fragile X phenotypes. The mice have less seizures, they behave, some of their behavioral impairments are corrected, the spine defects are corrected, and that work from Mark Baer, Kim Huber, and Steve Warren, and other colleagues led to this mGluR theory of Fragile X syndrome, which posits that exaggerated signaling through mGluR receptors that drives downstream protein synthesis might be corrected by antagonizing the receptor, and that could have a therapeutic outcome. So to make a long story short, a lot of this research in a number of labs in animal models, correcting the protein synthesis defect with mGluR5 antagonists, correcting other disease-relevant phenotypes, has led to human clinical trials. And now there are 3 pharmaceutical companies that are conducting phase 2 and moving toward phase 3 clinical trials with mGluR5 antagonists. And Emory is one of the participating medical centers. And next week, Elizabeth Berry-Kravis, who actually is at Rush in Chicago, she's meeting with the Fragile X Center here to talk about the results of a number of these ongoing clinical trials. I encourage you to go to her pediatric research grand rounds. Some of this was published in Science Translational Medicine on very interesting therapy. Actually, that paper it is on activating GABA receptors, but there's also a paper on antagonizing mGluR receptors. What Christina Gross in our lab has done is to ask, how does the absence of FMRP lead to exaggerated mGluR-driven protein synthesis? Is it because FMRP regulates mGluR? We don't think so. Actually, research from Nina's lab has shown that FMRP puts the brake on downstream components. FMRP doesn't regulate the receptor directly, we don't think. Her research shows that FMRP represses the PI3 kinase signaling complex that's tethered to the metatropic glutamate receptor. It actually represses PIKE, PI3 kinase enhancer. We're studying this in collaboration with Ko-Chung Yi here at Emory. And it suppresses the translation of P110 beta, which is the catalytic subunit of PI3 kinase. And then in response to receptor stimulation, this PI3 kinase is activated through the effects of PIKE to lead to downstream activation of mTOR and protein synthesis. So in the absence of the FMRP brake, this is where we think the molecular lesion is. It's an excess in the PI3 kinase signaling complex, its activity. And Nina indeed observed that as a robust phenotype in synaptoneurosomes from FMRP knockout mice, looking at PI3 kinase activity. And so what Nina is doing in our lab is to investigate whether antagonizing PI3 kinase signaling might be another type of therapeutic strategy. And in collaboration with Mika Nakamoto and Xiaodi Yao in Steve Warren's lab and our lab, a number of Fragile X phenotypes have been rescued by PI3 kinase inhibitors. This includes the dendritic spine defect. This includes the excess synaptic protein synthesis defect, rescued by PI3 kinase inhibitors. So we're moving forward, actually, working with subunit selective inhibitors from Glaxo. This is a whole new field in terms of PI3 kinase pharmacology. PI3 kinase is an important enzyme. You don't want to antagonize or inhibit all PI3 kinase, but there are a number of subunits, catalytic and regulatory subunits, and there's a number of companies that have developed subunit selective inhibitors that won't affect PI3 kinase function in a healthy cell, but might correct a dysregulated function in a disease cell. This study here done by Christina Gross, she envisioned that maybe patient cells, even their blood cells, would show this elevated protein synthesis. And this could be a useful, perhaps, a biomarker and maybe a type of assay that could be used for a drug screen. So in patient fibroblasts, in this particular assay, you're doing an assay for protein synthesis. And so you can see in Fragile X patients, in human patients, there's increased protein synthesis, similar to what we see in the FMRP knockout mice. And this can be corrected by this subunit selective P110 beta inhibitor in patient lymphoblastoid cell lines. This here shows that the drug actually decreases this excess PI3 kinase activity. So we're very excited about having a handle on a human population to study the signaling disorder. In the last couple of slides, how does this actually lead more broadly? Remember, I promised you that I would make some links between Fragile X syndrome, which is the most common monogenic cause of autism, with other autism spectrum disorders. So a number of monogenic causes of autism actually involve genes that act as brakes on this pathway. So Rett syndrome, neurofibromatosis, P10, tuberous sclerosis, a number of these genes that are mutated in autism regulate either directly or indirectly this pathway in either a positive or negative manner. So FMRP is a repressor. Beyond the single gene causes of autism, there are copy number variants that have been identified in many other forms of autism spectrum disorders, and the PI3 kinase downstream mTOR signaling pathway has been shown to be a hotspot for copy number variants. So we think that the work that we're doing on patient cells in Fragile X syndrome will have important implications. And we speculate that this regulated PI3 kinase mTOR signaling and protein synthesis might be a mechanism shared by many autism spectrum disorders. Mustafa Sahin from Harvard will give a Frontiers talk in April, and he is actually directing a clinical trial in tuberous sclerosis, a monogenic disease that's also a form of autism. And his clinical trial actually involves inhibitors of mTOR, which is downstream of PI3 kinase. And you'll hear about his work on a mouse model of tuberous sclerosis. In this last slide here, I'll talk about work that we're doing that's funded by the Autism Speaks Foundation, work that Ashwini Kupal and Christina Gross are doing to screen lymphoblastoid cell lines from monogenic forms of autism, idiopathic forms of autism, and also autism that may be due to copy number variants. And we have some patients that actually show some changes. One particular patient we're focusing on actually has elevated phospho-S6, which is a translation factor, a ribosome-associated protein that's downstream of mTOR in this PI3 kinase mTOR pathway. Very interestingly, this patient has strong elevation of phospho-S6, but it has elevation of P110 gamma P110 delta, and unlike P110 beta, which is affected in Fragile X syndrome. P110 delta is implicated in schizophrenia from work of Amanda Law and Danny Weinberger, and they are using P110 delta subunit selective inhibitors to correct for schizophrenia, at least in a mouse model. So I hope that I've given you an interesting twist on the RNA world and how studying RNAs and the associated proteins actually has important foundation for understanding neuronal function and its dysfunction in neurologic diseases. There are a number of people to acknowledge. Christina Gross and Wilfred Russell, who are assistant professors and have their own funded programs, and I collaborate closely with them. People whose data I talked about today include Paul Donlan, Marius Ifram, Ashwini Kupal, former lab members Claudia Fellini, Rami Matachetti, Yukio Sasaki, Christy Welshand, and Hong Lai Zhang. And I'd like to thank my Emory collaborators, Stephen Warren, Kachung Yi, James Zhang. Thank you, Yvonne Resnick, and a special thanks to Alexa Matisis from the Integrated Cellular Imaging, and collaborators at Einstein, Rob Singer. So with that, I'll end, and thank you for your attention.